Hi everybody, this is Maria Ruiz and I'm gonna talk about convolution modeling for the SPM course. One second, yes. So, this presentation is based on the original paper by Litvak and colleagues called Convolution Models for Induced Electromagnetic Responses. I include the reference at the end again. Now, the problem that the authors was trying to address, there were several problems, which are related to EEG and MEG analysis. In classical analysis of EEG and MEG, when focusing on events of interest, one would cut epochs around those events of interest and then average them. But that approach suffers from several problems. First of all, sometimes it's not clear where to place the baseline for normalization. The baseline might not be empty of previous events. And that's precisely the second problem. There is often overlap between consecutive responses, successive events, stimuli, etc., which have different latencies. There is also the problem that participants will produce different latencies when making button presses that contributes to variability in the signals, neural responses. And in more naturalistic experiments, we have a bit of a combination of all of these things, which results in us not really knowing how to form epochs, how long, etc. So to address these problems, the convolution model was developed. Today, we're going to see three examples of its application. One more detail, focusing on stop signal tasks. A second on decision-making from my own lab, and a third one where I will just briefly show you a kind of like a naturalistic, very rich paradigm where we can also use this approach. Now, in the paper by Jia and colleagues in 2015, they used this model for a stop signal paradigm. The question that the authors wanted to address was, what is the neural correlate of stopping a plant movement? And they recorded MEG signals. So in this case, we are going to see the details of the trials in a second, but basically what the authors usually would do with a classic approach would be to parameterize behavior, right? So you identify different aspects of behavior in that task, observing stimuli, observing cues, responding to them, making decisions, etc. And in this case specifically, to address the question, what is the neural correlates of stopping a plant movement, the authors wanted to isolate stopping specifically. And then one would analyze the corresponding neural activity. So as I said, for classic approaches in this type of paradigm, one would cut epochs around some kind of stop signal, average them, and extract the neural responses, for instance, in the time frequency domain. And one would conclude that is the MEG correlate of stopping. However, all of the problems we outlined at the beginning affect this task. We can see here a GO trial where after a warning signal, a fixation cross, there is a GO signal pointing towards the right or the left, and the participant has to press a response in the correct direction. Here we already have variation in the latency between the fixation cross and the GO signal, and the participant will produce the response at different reaction times every trial. In stop trials, after the fixation cross and a variable GO signal, there is a stop signal. And the latency difference between the GO signal and the stop signal is called stimulus onset asynchrony. This interval varies every trial, could be short, could be long, and depending on the SOA, the participant might be more likely to correctly stop the prepared action, therefore correctly stop, or if it's too late, if the fixation cross comes too late, the participant might be so advanced in movement planning that they do not succeed in stopping the action. So they actually produce a response. We can see this here as well. We have a lot of different latencies for fixation crosses, the same for GO signals, and then in stop trials, the stop signal cue, the red cross, appears at different latencies. Now, if this stop signal might be too late for the participant. They might produce an action. So this would be a trial where the participant, after the stop signal, doesn't successfully stop. Whereas in correct trials, the participant succeeds in stop. Now, here, if we would cut epochs around the event of displaying the stop signal, we are kind of like integrating into our analysis a lot of confounds. First of all, in error trials, you actually have responses. So there is a lot of motor preparation here that continues up until the response. This doesn't happen if the participant successfully stops the action. You also have different latencies between the previous cue signal, the indicating the direction of the movement, and the stop signal. So these events and these events have different overlapping properties with previous events, and that will reflect in the neural correlates. So if we just cut epochs around the stop signal and compare these trials with these trials, we would be including a lot of confounding modulations in our analysis. That's why the convolution model was introduced. An approach to that problem would be, first of all, to kind of like create a regressor matrix where every time you have the presence of a cue, for instance, the fixation cross, you code it as a kind of like some kind of like variable. So you have these dummy coded regressors that indicate presence or absence in black of a specific event for a specific regressor. For the ghost signal, you also have presence and absence of that ghost signal according to this display above. For the stop signal, you have also presence and absence coded in this regressor, and the same for the button press. Every time there is a button press, you denote its presence in this kind of design matrix. Here, PST denotes peristimulus interval, because you could define the presence of a stimulus or a cue around some kind of like window of interest, for instance, from minus 300 milliseconds to the actual presentation of the cue to 1 second afterwards. So you could define the presence of the stimuli in this peristimulus trial, peristimulus time segments, right? So this, in principle, could be any kind of like a first approach to a regressor matrix that then you could use to estimate the neural responses. From the time frequency domain of our signal, which we observe here, this is continuous time, and on the vertical axis we would have different frequency bins. We would, for instance, represent the whole continuous recording of EEG or MEG in this time frequency representation. This could be power or amplitude. In this paper, they use power. So you have the time frequency representation of your continuous signal. You have this kind of like a regression matrix, the design matrix, and then applying standard SPM, general linear model approaches, you could extract the regression coefficients for each regressor of interest. Now, in this simple approach, here we just illustrate the time continuous power representation of the signal for one frequency bin, and then running this GLM. In this simple case, we would obtain one time frequency representation first for one frequency bin, and then if we do this across all frequencies, we would obtain this kind of like time frequency representation of a specific type of event, the stop signal, for instance, around the peristimulus time. But what we can see here is that this peristimulus time modulation seems flat, right? So we haven't allowed this GLM yet to have any type of fine-grained analysis of changes over time during the peristimulus time. We have treated the peristimulus time as a flat present binary response, like either it's present with coding it as ones or is absent coding it as zeros. So this first approach is not really what we want. And by the way, this is in the classic general linear model. This is the residual noise. So we want something a little bit better. To do that, what we want to do is really like to capture how responses evolve over time, including over time around the events of interest. We want to construct regressors by convolving a set of basis functions with a set of input functions representing the presence or absence of those events. For instance, the presence and absence of those events could be represented by delta functions, something very precise in time, or stick functions. And as a basis function set, we could use functions that vary smoothly over time, like slow frequency temporal functions, to keep track of modulations of the signal during the peristimulus time. We could, for instance, choose a finite impulse response set, where here we can see what happens around the peristimulus time for different functions in this basis function set, or we could use a Fourier set with sines and cosines, going from low frequencies to a little bit faster frequencies. This is mostly what has been used in the application of the convolution model so far in the literature. This is also what they use in this paper and what we have used as well in our own work. Each basis function gets its own weight, therefore allowing the model to capture the dynamics of the induced response over time, peristimulus time, and frequency. Here we have the representation. Again, the basis functions in this illustration can be observed here, going from lower frequency to higher frequency basis functions. These are the beta coefficients, in this case, for one specific event type or regressor. And for every, so one would extract the beta coefficients for each basis function and for each frequency bin, and then for each event type of interest or regressor, right? So stop signal, go signal, fixation cross. Here we are illustrating what would be the convolution modeling approach for just, for instance, the stop signal. So after the convolution modeling, we would get these so-called images, these time frequency representations of the induced response to this specific regressor, and using, after having collapsed information across all the basis functions. As you can see here, now we have a rich representation of changes across frequencies and during the peristimulus time. So, going from our original first approach of a GLM, where we just had presence and absence of stimuli and cues in a binary representation, we now would have a representation of the presence of the events as, for instance, ones only when they are present at that specific latency, very precisely located in time, but now we allow variation by convolving these kind of like input functions with the basis functions, right? And through convolution of the input functions with the basis functions, we obtain a richer design matrix, which we can also see in this representation. Now you can see that every time that a specific event of a specific event type is present, we have this rich representation of its presence throughout different basis functions. So this would be our updated design matrix, right? And we can also include elements for baseline drifts and other kind of like regressors that we can include in the model, as you would do for fMRI analysis as well. So here, basically, we have two representations. First of all, we have our original time frequency representation of the continuous signal, or the signal Y we want to explain, which again could be power or amplitude. Here we have the this rich design matrix, where here you can see for each regressor, which would be a column for each time that there is an event present for that specific regressor, you can recognize here the basis functions, and here could have this kind of like baseline drift. And the aim of the convolution GLM will be to use the SPM machinery to estimate the beta coefficients for each of these different elements, for each frequency band, for each regressor, and for each time, and for each basis function throughout the peristimulus time. Now, in this representation, we see the beta coefficient matrix and the design matrix, but we can just extract, for instance, one specific column here representing one event type, let's say the stop signal, and here we can see how the specific basis function for a specific regressor, once they are combined with a specific beta coefficients for that regressor or event type, would give us the specific time frequency image for one specific event type. Again, let's say this could be the stop signal event or the go event, etc. Obviously, for understanding the maths and looking at all the possible matrices and their indexes, etc., go to the original paper where everything is explained in detail. Now, going back to this paper by Jia and colleagues, they used this approach, and what they found was that the images, time frequency images that they obtained from the GO signals across different motor areas in the primary motor cortex and supplementary motor area and pre-SMA and in the inferior frontal gyrus, they found different time frequency modulations for this GO signal. They also found different time frequency responses to a button press. As you can see here, we have some kind of like increased beta power after the button press as expected, right? And the interesting thing is that since you're modeling all of these events or regressors simultaneously, when you look at the specific neural responses to the stop signal, you are already accounting for any other modulation by these other regressors, right? So you are explaining away every the neural response to every regressor, which allows you to focus then on a specific neural response for one regressor of interest. So this is the same in a different representation, right? The original time frequency analysis, the time frequency images for each kind of like event of interest for the fixation cross, go signal, stop signal for different participants. And then this is called the first level convolution model, conducted for every participant separately. And then you can, for instance, look at the go signal and obtain these time frequency images for each participant, average them, and then do some plots. Or, of course, you can not average them and use them for statistical analysis, right? This would be a standard second level analysis. You can use your standard statistical test here. And then, so here they looked at, for instance, the successful and unsuccessful stopped trials, so stopping trials where the participants, after preparing a movement, they managed to successfully stop or not stop. So here the authors show a statistical contrast between successfully stopping and non-stopping in stop trials, and they look at the statistical effects in these different motor areas and in the inferior frontal gyrus. So as you can see, there is some kind of like significant effect in this contrast, specifically in the beta band, around 200 milliseconds to 900 milliseconds after the successful, after the stop signal. So you can see this robust effect here in the statistical maps, more prominently for the pre-supplementary motor area and in the left inferior frontal gyrus. You can see other effects there. So here the authors successfully identified the neural correlates of stopping in this task while having accounted for all of the other modulations of neural responses by the other explanatory regressors. Again, also taking into account different latencies between different cues, different response times by participants, etc. And this is also a different representation of these effects that they found. So overall, they could then conclude that successful stops were associated with a beta increase in more frontal regions. Now, this might seem like a result that obviously is meaningful, it makes sense, but it could be confounded if we would have done the classic epoch average approach, right? Perhaps also those classic approaches lack sensitivity. Sometimes it's not so much related to whether you get opposite effects, is obviously more related to the sensitivity of your analysis. And sometimes you can get confounding factors contributing to your effect of interest if you use the classical approach. A second example is from our own work. For instance, we've used this convolution model a few times. In this paper by Thomas Hein and colleagues, we were interested in analyzing the neural correlates of decision-making under uncertainty in individuals with different levels of predisposition to trait anxiety. So we were looking at how anxiety modulates decision-making on a behavioral level and also the neural correlates of that. Now, the reason that I show this as an example is that we have to deal with some of the problems that were outlined at the beginning. We have a task where participants see some stimuli, and after a varying time interval, they will choose which image they prefer. Here they have to identify which fractal is more likely to be rewarded. So participant makes a decision. The reaction time is going to be different every trial. So here is one of our confounds. Also, the stimuli will be presented with some jitter trial after trial. Once they make the response, the participant will observe again with some jitter whether they made the right choice and they receive a reward or they don't. And then there is some inter-trial interval with jitter as well. So here we were interested in understanding the neural correlates of updating the beliefs about which of these fractals is more likely to be rewarding. To do that, we use a computational model of behavior, of decision-making behavior. I'm not going to go into details, but the important thing for this presentation is that when using computational models of decision-making behavior, you can extract parametric variables that, for instance, code prediction errors that reflect the mismatch between expected and observed outcomes. And in this case, we use a variable which was a continuous variable, which is termed precision-weighted prediction errors. So these prediction errors are weighted by the precision of the belief distributions of the participants. And basically, for the sake of this presentation, we're just interested in emphasizing that these so-called variables of interest are parametric, right? So they are continuous variables. They increase, they decrease, they could be positive, negative. It depends on the variable, computational variable of interest. So what we did is we applied this convolution model by defining kind of like an input function with discrete regressor, the presence and absence of stimuli, right? Like stimulus presentation, response by the participant. Every button press was a discrete regressor. When they observe the outcome, that's another discrete regressor. You would have two discrete regressors in this case, whether they win or whether they lose, but also discrete regressors for responding left or right, for instance. So you could design your input functions using as many regressors as are relevant for your task, but we also wanted to include parametric regressors, which is something that the GLM, this convolution GLM allows you to do. You could combine discrete regressors, as in the previous example, with parametric regressors. And therefore, this will allow you to identify the neural correlates that are parametrically co-modulated with a specific continuous signal. So we wanted to, for instance, identify the neural correlates of precision-weighted prediction errors in the time frequency domain while accounting for discrete regressors in parallel. So this is kind of like a similar graphic to the one you saw before. In this case, you see time frequency representations of our signal. This could be the continuous MEG recording, or you could select segments which include your regressors, your events of interest, and you could concatenate these segments. This is called pseudo-continuous signal, and both approaches work well. And then we would create our design matrix with, for instance, discrete regressors and a parametric regressor at the end. And as you saw before, each presence of these events would be represented by a basis function, and then through the convolution modeling, you could obtain a time frequency image giving you this fine-grained time frequency response to a specific regressor. And in this case, this could be a parametric regressor or a discrete regressor, etc. And then you could obtain this for each participant, and then you could obtain statistical maps. We did that in this case using source localized time frequency analysis, and we applied the convolution model in the source domain, and we found that there were some reductions in alpha and beta activity and increases in gamma activity in parts of the anterior cingulate cortex and the prefrontal cortex that were sensitive to changes in updating beliefs between participants with high and low trait anxiety. And these neural findings were aligned with the behavioral effects that we've also found in terms of like participants with high or low trait anxiety updating their beliefs faster or slower in this task. And as I said, at the same time, we were accounting for confounding factors such as the responses, right? So every time the participants press a button expressing the response, we would also have that as part of the convolution model. So we could explain away the classic expected findings like suppression of alpha and around the movement and beta rebound after the movement. So we explain away all of these modulations to other regressors, which allows us to isolate the neural responses to the regressor of interest, which in this case was this parametric continuous variable term precision-weighted prediction errors. And to finalize, here I'm just going to show you a type of naturalistic paradigm, where by naturalistic we mean a very rich, highly complex task, which is playing video games, where we had a study where participants played a real video game on Steam. The game is called Outriders, and in this task, participants have to do thousands of events, a lot of button presses from different types of buttons in the game controller. We represent here in one participant throughout time how many events of each type we would have. For instance, we would have discrete events every time the participant presses a button across each of the 8 buttons of the game controller, right? So you see there are a lot of discrete events across different types of button presses. We have events, discrete events, for instance, when the participant kills somebody else, that we call that X-death, so killing others. The participant sometimes might die. These are the brown crosses. These are obviously very salient events. There might be only 5 of them, but we want to code for them as well. And then we would have parametric events such as health. Your health level could increase or decrease across a continuous scale, and your score level, the reward points you get, also increase over time. So we successfully applied the convolution model to this type of data, and it works quite well. It's not necessarily much more computationally expensive. It's solved quite successfully within a good time, and we have had some very interesting findings, but I'm not going to show them here. I just wanted you to see how in naturalistic settings where you have many things happening in real time, you want to code for all of them that you identify as relevant ones, and you can model the neural responses, whether EEG or MEG, to each of these regressors in parallel. Now, you will be able to run these convolution models either through batch scripts, there is available code, or you could just run them using the SPM toolbox. I personally prefer to run this in code. You will have segments of code to define discrete regressors, but also parametric regressors. And I believe that you will have access to code to run some examples that the SPM team has provided for you. So to summarize, we outlined some of the main problems of EEG and MEG analysis when dealing with epochs around events. And those problems related to the epochs often not having a very clear baseline period for us to kind of like normalize the data. There is also the issue that we often have overlapping neural responses to stimuli or events that are close in time, and how close they are in time varies trial by trial. We always have different reaction times from humans, right? So our reaction times have variability. The variability will be dependent on each individual, but also on their own throughout the task changes, right? In fatigue, in cognitive load, in many other factors, confidence, etc. So convolution models for induced responses solve this problem. They allow you to obtain time frequency images for specific events of interest while explaining away the neural responses that are related to other regressors. There are many advantages. You can include discrete and parametric regressors. Some of interest, some might be nuisance regressors. You can model everything at once, and it will allow you to really address your scientific questions of interest while you are getting, you are trying to control for confounding factors. Here we have the main original paper introducing this method with the maths. Have a look at it, and the examples that I covered are this one by Jean colleagues, one of our own studies using this model. We've used it a few times now, and I haven't covered this approach by Spitzer and colleagues, but it's also quite relevant, and I believe that you will also have a code to kind of like implement this approach here.